And as you can see from the slide, my topic is evidence for variation in human radiosensitivity, potential impact on radiological protection. So I intend to go through some of the strands of evidence for variation in human radiosensitivity, and a lot of that work will be based on a report that was published in the UK by the Advisory Group on Ionising Radiation a couple of years ago in 2013. But I have to say this talk is very much my personal view that does draw on that report. So we know in the current system of radiological protection, it serves to avoid tissue injury, or so-called deterministic effects in the old parlance, and also minimize the risk of stochastic effects. And those primarily are cancer, but also a contribution from hereditary effects. And this is a population-based protection system, which is based on dose limitation, and the dose limits are derived from a notional average that doesn't really exist. You'll recall earlier on there was a discussion in the radioecology session from work package 5, from committee 5 about the different sort of ellipsoids that can be used to describe different organisms. There's variation there. And there was an example, I think, yesterday of how height and body size, body shape can affect the doses that people receive in a medical setting. So there is variation amongst us, and we know that we are all different. Look around the room, we are clearly all different. We're different in terms of our lifestyles, we're different in terms of our genetics and so forth. And that can be seen very obviously in radiation risk. When you look at the gender-specific differences in risk that have been mentioned at several occasions in the past couple of days, and you can see that clearly in the breast cancer risk, where the excess relative risk is very much more for females than it is for males. So we know we're different, we know there's variation. And a lot of variation can be described by a number of different endpoints, and when talking about radiosensitivity, one has to be very clear about defining which endpoint of radiosensitivity you're talking about. Perhaps some of the most clear evidence and the most abundant evidence comes from the clinical setting, where patients are exposed in the radiotherapy setting, and in some instances there's adverse normal tissue reactions to that exposure. And this is a figure taken from the AGIR report I mentioned, looking at a cohort of some over 1,000 breast cancer patients, looking at the range of normal tissue reaction, effectively described by this measure residual score, where you can see there is clearly a spread with a clear median, but there's tails to either side showing populations with very much increased sensitivity to those normal tissue reactions on the right-hand side and somewhat reduced radiosensitivity on the left-hand side. Thinking about the factors that affect that range of sensitivity, it's known there's a number of individual factors that do come into play for normal tissue reaction in radiotherapy. Increasing age clearly plays a role. Smoking behavior plays a role. The presence of other conditions like diabetes or collagen vascular diseases is known to have an impact, as well as genetic factors. And there's not really sufficient evidence to draw conclusions on the role of sex, ethnicity, body mass index, diet, alcohol, etc. So we know some of the individual factors, including genetic factors, do affect clinical radiosensitivity. That is the induction of normal tissue reaction following radiotherapy exposure. We also know that there's a range of radiosensitivity syndromes within the population. These are quite well described, and these are generally rare, when I'm talking 1 in tens of thousands of births usually, rare recessive disorders leading to cellular and occasionally clinical radiosensitivity. There's a range of syndromes for sure, a number of them are listed here. Ataxia telangiectasia is perhaps the archetypal one. There's also Fanconi anemia, Nijmegen breakage syndrome, Cornelia-de-Lange syndrome, perhaps more recently described, and severe combined immunodeficiency syndrome. So we know there's a number of syndromes, a number of individuals within the population out there that are showing very substantial radiosensitivity. The basis of some of those syndromes, at least, is within DNA repair pathways. The so-called non-homologous end joining pathway of double-strand break repair is involved in a couple of them, and also the homologous recombination repair pathway involved in Fanconi anemia, for example. DNA damage signaling, so the way the cells respond are important in the ataxia telangiectasia, Nijmegen breakage syndrome, and another syndrome known as Riddle syndrome, which I haven't actually mentioned yet. And also another factor, sister chromatid cohesion, the way that the DNA and chromosomes are structured and organized within the cell can play a role. And a couple of syndromes there, Cornelia-de Lange syndrome and Roberts syndrome, are known to be involved, known to have an involvement of that cystochromatid cohesion mechanism. Turning now to consider cancer rather than radiosensitivity per se at the cellular level, we know within the population from pediatric syndromes, pediatric cancer-prone syndromes, that there are sensitivities to radiation-induced cancer after, again, therapy for primary cancer rising spontaneously. In the retinoblastoma syndrome, where you have eye tumors, there's frequently soft tissue sarcomas found in radiation fields. In neurofibromatosis type 1, second cancer associated with radiotherapy for gliomas, which occur in the initial patients. Liefeld-Meany syndrome, which is deficient in the p53 pathway. There's a high relative risk of second and even third cancers following radiotherapy. And in Gorlin syndrome or basal cell nevoid syndrome, there's multiple skin cell tumors, skin cancers that occur within radiation fields. So there is evidence within the childhood population, again with a strong predisposition to cancer already, that there is predisposition to radiation-induced cancers or radiation-associated cancers as well. So that's children. What about adults? Adults are important as well, and there's some studies from breast cancer studies looking at specific syndromes with known breast cancer susceptibility and the impact on those of radiotherapy on cancers arising in those individuals. And the classic one is the ataxia-telangiectasia syndrome, again, ATM heterozygotes, that's people not with the syndrome itself, but carrying one faulty copy of the ATM gene, if you like. And ATM is a gene which is responsible for ataxia-telangiectasia within the population. There's some studies of variable quality indicating elevated risk, approximately twofold risk in this particular study. Another study, which is perhaps somewhat more robust looking at demonstrated greater risk of contralateral second breast cancer in those who've had a first breast cancer and a second cancer occurring post-radiotherapy, again, in the second primary there. And another study indicating a few other genes, perhaps some association. But I'd like to note again that these studies are variable in their quality and their robustness, and that the middle one there is perhaps the best and most reliable one can find in the literature at the moment. So this is generally suggesting that finding the genetic factors that underlie radiation cancer susceptibility is a pretty big challenge. And I'd just like to outline to you a couple of studies, or highlight to you a couple of studies that show really quite how difficult it really is. So part one, the WeCare study, a large study of some 50 or more than 50,000 women with breast cancer. And within that study, there's a nested association study looking at 700 odd contralateral breast cancers. That was the second breast cancer occurring within an individual who's had a primary breast cancer spontaneously, with some 1,300 controls. So these are, again, we're looking at the post-radiotherapy cancers arising in those who've had a primary breast cancer and the second breast cancer in the contralateral breast occurring after that initial therapy. And the WeCare study, as has looked, and this involved John Boyce amongst others in a large consortium, looked at a variant of a CHEK2 gene, a cell cycle checkpoint control gene, where there's some indication of increased relative risk, but it wasn't significant. There's the ATM study I mentioned in the previous slide, which has one reasonably strong association at a 2.8-fold increase in relative risk after the radiotherapy exposure. Looking at 100-odd SNPs in 6 ATM target genes, no associations found. BRCA1 and BRCA2 variations, no associations found. SNPs in other breast cancer loci, no associations found. Pregnancy factors, so not necessarily genetic per se, but there's one strong association with the increased relative risk. So that showed there was some positives, if you like, but not all of them by any means. A second study, not looking at radiation specifically in this case, but a very large breast cancer study looking at some environmental risk factors in 35,000 odd breast cancer cases, 35,000 breast cancer cases and 40-odd thousand controls. Looking at 22 SNPs, so single nucleotide polymorphisms, these are variant genes, if you like, and looking at the modification of risk by those variants by 10 established environmental and, as I said, non-radiation risk factors, but again, a demonstration of the difficulty in finding, or the challenges in finding associations. Some associations were found, for sure. Increase with one particular gene of a modest size with increased parity. A link between alcohol consumption and one specific gene there, again a fairly modest increased risk, and one final one with a relatively strong association, again with parity. So there are certainly some associations, but the number of potential risk factors, genetic risk factors that could have been identified in this study is really quite substantial when you're looking at 23 variants and 10 environmental factors. But the fact is, there was a relatively few that have actually been found and shown to be statistically robust. So it is actually quite difficult to find these things. And that was in studies looking at therapeutic exposures, so high-dose exposures. What about the diagnostic setting, which is perhaps much more common form of population exposure, that's for sure. And there are some studies, again, in the literature out there, BRCA1 and BRCA2 carriers. These are BRCA1 and BRCA2 are the best described breast cancer risk factors we know about. They've been suggested to have increased risk of breast cancer following multiple chest X-rays. And in mammography setting, there's mixed evidence available within the literature. I've not listed the studies here because there's quite a few, but on the basis of what's known about breast cancer and radiation risk and the age association and the frequency of screening there would be amongst individuals, amongst females through mammographic screening programs around the world, it's been suggested that screening from 35 years of age and on is potentially beneficial, but at younger ages it certainly wouldn't be. And that's from Amy Berrington and colleagues a few years back. So, what would the clinical implications of these current level of information actually be? We know that normal tissue damage limits the ability to control tumour growth, and so if one could predict radiosensitivity of those individuals with cancers, that would help tailor the treatment to those individuals, and certainly advice to individuals on potentially modifiable risk factors, and smoking and alcohol consumption would be some of those potentially, would be of real benefit to help optimize the benefits of radiotherapy. And that sort of echoes what I think Jolene was saying earlier on, that the factors that affect baseline cancer risk can also affect post-radiation risk. Furthermore, increasing use of medical diagnostics, especially CT, could pose a few problems for those really highly radiosensitive individuals within the population, those with the syndromes, the ataxia-telangiectasia, Nijmegen breakage syndrome, etc. Now, those are relatively few in number, those syndromic patients, if you like, and they're generally identified, but not always identified. So identifying such individuals within the population would be really beneficial and help find them before they're actually exposed to any radiation, be it diagnostic or therapeutic. And of course, one needs to maintain the justification for any medical diagnostic procedure on the basis of the clinical benefit derived from using that procedure, outweighing the potential risk there is from the radiation exposure itself. And that sort of area has been discussed quite extensively yesterday. And don't forget that interactions between factors can be important. We know about the smoking and radon interaction that's very well characterized, whereby the majority of lung cancers are rising within radon-exposed populations, or high radon-exposed populations, to high background radon levels, occur within the smoking subfraction of those populations. And also remember that exposure to radiation alone in the medical setting is actually very, very rare. So the majority of cancer patients, at least, will be receiving radiotherapy in combination with chemotherapy or other therapeutic interventions as well. So you rarely have the opportunity to identify radiation sensitivity in a pure radiation-exposed population. And indeed, the knowledge of that interaction might be important in determining how one handles risk and risk communication, risk evaluation in those contexts. So how are we going to move on to use this kind of knowledge we have at the moment? We know there's some level of variation in radiation sensitivity. We can characterize some of it, but only probably rather a small portion at the moment. And I would argue that the ability to identify those individuals who are sensitive will be the key factor. So reliable prediction would be key. And there's potential for genetic testing, which is mentioned in some of the genetic screening type approaches, or there might be cellular and molecular endpoints or readouts that might be informative as well. But that will certainly require the accuracy of prediction speed to allow quick judgments to be made in a clinical setting, reproducibility, obviously it has to be a reliable test, and acceptance that it actually brings benefit to the individual and to the population as a whole. So these are really quite a high hill to climb, I would argue, that you need to have very strong and robust assays before you apply them in any sort of practice. So there are a number of activities looking for genetic predictors of radiation therapy reaction. I'd like to highlight the Requite study here, which is recruiting bloods from cancer patients undergoing radiotherapy. This is coordinated by Kathy West in Manchester. The results are far from in, but the recruitment process is underway. And this program may well identify some genetic predictors of acute normal, I'm not sure if it's acute, but acute and late normal tissue reaction post-radiotherapy in large international cohorts again. So again, large studies are needed to identify these risk factors if indeed one can. But as well as genetic assays, as I said in a previous slide, there's probably other readouts that one could use, and perhaps more readily, of a cellular or molecular nature. I'd like to highlight a couple of those here. One for radiotherapy reactions, work done in France by David Azria and colleagues notably, looking at apoptosis induction of a form of cell death in T lymphocytes exposed to 8 grays in vitro. It has been found to be predictive of late normal tissue reactions in 400 odd cancer patients. And I believe that's being used further in that clinical setting. And also perhaps at diagnostic exposure levels, there's indications from studies from Nicolas Fauré's lab, again in France, that looking at DNA damage foci within cells, the gamma H2AX foci, which are sort of points within the cell nucleus where repair functions are coordinated and aggregated, after low dose exposures now, 2 to 4 milligray in mammography type X-ray exposures in mammary epithelial cells, more of those DNA damage foci were found in patients with high breast cancer risk, and that was based as at least 20% risk based on family history or the known mutation carrier status. So there are indications for sure that cellular assays might be predictive of risk in these particular settings. I'd like to highlight a couple of areas of work which is not in any clinical setting at the moment, but work from colleagues at PHE looking at other potential markers. And this study looks at a specific gene and the expression of that gene, CDKN1A, which is a cell cycle regulator, which appears to be associated in a very small study. I have to admit, this is not a large study of breast cancer patients looking at severe early radiation toxicity in the skin. And the relative gene expression after radiation exposure here is plotted against the individual patients and ranked according to that relative gene expression level after radiation exposure. So this is 2-gray radiation exposure and looking a couple of hours after that exposure in an in vitro system, again in blood samples from these individuals. And you can see, because the normal reactors are highlighted with red points here and the severe reactors with the blue points, there is apparent distinction between those two groups in their relative gene expression after radiation exposure in blood cells and their normal tissue reaction status. And you can put in a admittedly arbitrary cutoff point, which will give you a reasonable prediction of their likely normal tissue reaction status, normal skin reaction status acutely after radiation exposure. That's again cellular radiation, normal tissue radiation sensitivity, really perhaps for more general application, having a reliable way to predict cancer susceptibility or cancer risk after radiation exposure would be most useful. And this is very much a proof of principle type experiment I'm going to run through here. But looking at one of the central pathways within DNA damage signaling, you'll recognize some of these names from the previous slides of the genes involved within the radiation sensitivity syndromes, the ATM pathway, and the critical factor here, P53, a gene which is most commonly modified in all human tumors studied to date, and eventual downstream targets, P21, puma, and cestrin, which are genes of the expression of which you can quantify very accurately in so-called quantitative PCR assays. Using mice, and mice are good because you can do all sorts of genetic tricks with them, you can play around with a number of copies of the P53 gene. So you can actually abolish it, so have 0 copies. You can have 1 copy, which is obviously reduced in number, or you can actually get so-called super P53 mice with multiple copies, so up to 4 copies of that P53 gene, and ask the question, what is the impact of the P53 gene number on the gene expression level of these downstream readouts, if you like, on gene expression? And I'm not going to go through the detail because it's a little bit too complicated, but basically, to the right-hand side of the slide, you're seeing the gene expression readout here the fold change in gene expression for P21, puma and cestrin, these 3 P53 responsive genes, plotted against P53 gene copy number. So you've got from 0 to 4 copies of the gene. With more copies of the gene, you're getting a stronger response post-irradiation, again 2 gray irradiation, and a couple of hours after exposure, again in T lymphocytes. So the P53 copy number is controlling the strength of response of these genes to radiation. Now that's interesting in itself in some ways if you're a molecular biologist, but it's perhaps more interesting if I tell you that the P53 copy number also determines lifetime cancer risk in these animals as well. So you can then plot P53 copy number, which you can do against lifetime cancer incidence. And what this allows you to do, because you've got this relationship between increasing copy number, giving you decreasing lifetime cancer risk, you can change the P53 copy number axis on the gene expression readouts to fold change in gene expression, which you can actually measure readily in any individual or any sample. So the cancer incidence is related to that post-radiation P53 gene expression response. So P21 levels, when you have a high fold change post-irradiation, you're getting a low cancer incidence. And with puma, the same, high fold change in gene expression, but lower cancer incidence. So there's a linear relationship between cancer incidence occurring through lifetime. So there's an early readout, appears to be an early readout of lifetime cancer risk in these mice. Now, I don't want you to go away with the idea that this is a test at all nearing clinical application, because it certainly is not. It's very much a proof of principle study, suggesting that there might be readouts that can be readily obtained at relatively short times after exposure that do relate to lifetime cancer risk. An awful lot more work will be needed in this area to bring it anywhere close to clinical application. But I think there is some hope that it might be possible at some future date. So there's not only scientific concerns, but there's also ethical concerns within this area. The ability to identify sensitive groups and individuals will need careful consideration, especially when you're talking about occupational situations. And we need to consider the justice of protecting potentially small numbers in the population at very high risk, or the justice judgment there is important. And in the absence of routine tests for sensitivity, provision of information on risk modifiers to individuals and risk modification strategies and the reduction and avoidance of smoking and alcohol consumption, for example, should be the main focus. So informing patients about what they can do to help themselves. And we don't know what the illegal implications of being able to identify radiosensitive individuals within the population might be. That's something to consider at some future time. The requirements then will be to have a good knowledge of the range of radiosensitivity within the population. We will need to have very reliable methods to predict that sensitivity. We'll have to have an agreement on a consensual basis that there will be benefits to be gained through being able to identify sensitive individuals within the population. And one will need to develop some sort of robust framework in which to operate, so to apply this knowledge and to apply it in an equitable fashion in a radiation protection context. And finally, obviously, you need to have the broad acceptance that is a useful and constructive thing to do. So the move from population-based protection to individual-based protection does require these things to be worked through carefully and not only at the scientific level, but also in terms of the ethical implications and the acceptance by a wider population. So, just to finalise, as I say, thank you very much for your attention. And here is a picture of the front cover of the Human Radiosensitivity Report, published under HPA, which is the precursor organisation to Public Health England, and it's available free to anybody at that website address there. So, thank you very much for your attention.